Hi everyone, so today we're doing DNA. We're going to focus on the introduction aspect of it, meaning looking at your structure of DNA and RNA in terms of the nucleotide and their molecule structure. Right, let me just do my notes so I can be able to remind myself because this is revision time. Okay, so I am doing the intro for DNA code of life. Okay. Right. So with this one, I need to first understand that we've got a nucleic acid, right? Which we also learned in grade 10, but now it's much more emphasized. So a nucleic acid is a type of organic compound. As I said, you learned it in grade 10. And with grade 10, you were also learning about even other ones like water and all of that. So here we'll just focus on this one, the fact that a nucleic acid is a type of organic compound. I don't have to go deep into that. Okay, and then we remember that there are two types of nucleic acid. There are two types of nucleic acids. Okay. Now, these notes of mine, I like putting color, and if you feel like you're interested in them and you want to get some, you can just let me know and I will hook you up. Right, so with the two types of nucleic acids, the first one is DNA. I never like to just use the abbreviation DNA. It's deoxyribonucleic acid. So there is your D right there going here. There is your N here and then your A right there. So it's much preferable if you use the full word. And then you also have RNA. Also, then it would start from the R. So this one would be ribonucleic acid. And please, guys, I'm probably going to pronounce these words wrong, but for me, emphasis is on writing them properly. I don't want to get my spelling wrong now. Okay, so nucleic acid is a type of organic compound, and now we understand that there are two types of nucleic acids. It's the DNA and the RNA. Now, these ones, they just form the basis of life, right? It's probably not important, but just to get the fact that they are in our bodies, they are important. Just like when you eat something that has potassium, and it's also important. It builds your cells and all of that. So they are just the basis of all life on Earth. Okay, and then most importantly, then we need to understand that these nucleic acids of mine are made up of building blocks. Okay, they are made up of building blocks. And then this word building blocks, they like to use the word monomers. So when you refer to building blocks, you are referring to monomers. So they are made up of building blocks called nucleotides. I know all these words sound so familiar, but that's just the nature and the interesting part about it. Okay, so we recap. With your DNA topic, you need to understand that you've got a nucleic acid, which is a type of organic compound. There are two types of nucleic acids, DNA and RNA. And then these are the four words you must learn: deoxyribonucleic acid and ribonucleic acid. They just form the basis of all life. Most importantly, they are made up of building blocks, monomers called nucleotides. Right, let's get now the basic structure of a nucleotide. Basic structure of nucleotide. Okay, I'm going to use my 3 colors for that one. The first one, which is always there, always, and I noted myself saying always, there in circular form is your P, which is your phosphate group. Okay. And then you've got your sugar molecule. Usually it's referred to as pentose sugar just because of this shape here. 1, 2, 3, 4, 5. But it's fine if you just say sugar. And then we have our nitrogenous base. Okay. Let me put right there. Okay. So this is just a basic structure of your nucleotide. This is what one nucleotide or one monomer, one building block will look like in order for you to form your DNA or your RNA. So this is the basic one. Okay. So just to put down there that the P is a phosphate group. Okay. Then the S stands for your sugar molecule, and then the NB stands for nitrogenous base. Nitrogenous base. Okay, I like to remember this as please stop now, babe. Like, I don't like what you're doing, so just please stop now, babe. Just stop. Okay, right. Just a quick recap of your notes. Again, with DNA, you must learn what a nucleic acid is, which is just a type of organic compound. There are 2 of them. It's DNA, deoxyribonucleic acid. It's RNA, ribonucleic acid. Then they just form the basis of all life. Importantly, they are made up of building blocks, monomers called nucleotide. And then here is just one nucleotide. It looks like that. It's got a phosphate group, a sugar molecule, and a nitrogenous base. Please stop now, babe. Right. Okay, so here's my nucleotide. Now I'm going to do a table that differentiates the 2 nucleic acids, right? All right, so let me draw that table of mine. Okay. Not too fancy. Just trying to remember what is DNA, what is RNA. Okay. DNA. Okay. RNA. Right. First thing I need to know about their differences is their sugar molecule. Their sugar molecule is different. There was my pencil there. The one thing they are first different in is the sugar molecule. Okay, so we need to get to those ones now. The sugar molecule for DNA is deoxyribose sugar. Then the sugar molecule for RNA is ribose sugar. It's almost like nice because now you remember that, okay, firstly, DNA full name is deoxyribonucleic acid. Then the sugar molecule for DNA is deoxyribose. So just there. Then RNA is ribonucleic acid, and the sugar is just ribose sugar. Okay. And the next thing they're different in is their nitrogenous bases. Remember when we did the nucleotide, they both have a phosphate. It's right there, not a problem. Then when it comes to the sugar is also here all the time, but the sugar for DNA is deoxyribose. Then the sugar for RNA is ribose, which we just did now. Then the next thing they are different in is the nitrogenous bases. All right, so we're going to list those ones out. Okay. Nitrogenous bases. My handwriting is nice, I know. Okay, the nitrogenous bases that they have, I'm going to color them in, but I want us to list them first. The first one is adenine. Okay, they both have adenine. They both have that one. Okay, then the second one is thymine. Now, DNA has thymine, but RNA has uracil. Okay. The third one they both have is cytosine. They both have cytosine. The fourth one they have is guanine. As I said, my pronunciation is for me to remember. Okay, so let's go through that. I'm going to be using colors to emphasize the differences and the similarities. Right. So DNA and RNA have the first nitrogenous base similar, which is adenine. So I'm going to use the green for that one. It's the same, AA, not a problem. Okay. The second one that they have different now is that DNA has thymine, where else else RNA has uracil. So that one is different for them. Okay. Then they have cytosine is the same, so I'm going to use my same color. Oops, where's this one gone? Okay, cytosine is the same. Then the last one, which is the same also, is guanine. Guanine is the same. Okay. Something I want to emphasize here with the nitrogenous bases, the now babe, is that for DNA, adenine works with thymine. So it pairs with thymine. All the time, adenine will pair with thymine. And the reason my arrows are like this is because it even works vice versa. If they start with the nitrogenous base thymine, then it will be complemented by adenine. Even if they start with adenine, it's complemented with thymine. Same goes with RNA. RNA with the adenine, it always pairs with uracil. So if it's an RNA strand, as I'm going to show you, adenine always pairs with uracil. Okay, then we come back here with DNA. Cytosine is always pairing with guanine and vice versa. Same even on this side. Right. And then the last thing they have differently is a strand, which I'm going to show you just now. But let's just write the difference. So the strand molecules that they have and how they are placed is that DNA has a double strand. Okay, see that D going right there. DNA, deoxyribonucleic acid, deoxyribose sugar, double strand. RNA, ribonucleic acid, ribose, but then it's a single strand. Okay, so as I said, I'm going to get the pictures with the strands molecule. DNA, deoxyribonucleic acid, deoxyribose sugar. The strand molecule is double-stranded. This is what we mean when we say double-stranded. Here is a single nucleotide right there. There's the phosphate group, the sugar molecule, and a nitrogenous base, thymine, which pairs with adenine right there. Okay, so here's the double strand. It's got this one and that one. And these ones are joined by your weak hydrogen bonds. The reason they should be weak is so that when we go to DNA replication, it can be able to unwind. Okay, we're going to go to that in the next video. So just remember, DNA is deoxyribonucleic acid. It's got a deoxyribose sugar. It has a double strand, and here's the double strand. With the double strand, one nucleotide of adenine will pair with one other nucleotide of thymine. Then one nucleotide of guanine will pair with another nucleotide of cytosine. And these nucleotides are paired by weak hydrogen bonds. Right. With RNA, on the other hand, ribonucleic acid has a ribose sugar. It has a single strand. There's the single strand. It's just that one. Right. Okay, so one nucleotide of adenine followed there by another nucleotide of guanine in whatever way it pairs itself. All right. Okay, let's do a quick recap of our notes. Our topic is DNA code of life. I need to know what is a nucleic acid, and a nucleic acid is a type of organic compound. There are two types of nucleic acid. It's DNA, which is a deoxyribonucleic acid. It is RNA, which is ribonucleic acid. They form the basis of all life on Earth. Most importantly, they are made up of building blocks called monomers. And a building block or a monomer is referred to as a nucleotide. Here is the basic structure of a nucleotide. It's got a phosphate group, it's got a sugar molecule, it's got a nitrogenous base. P phosphate group, S for sugar molecule, and NB for nitrogenous base. Okay, and then when you do now a differences of the two with the DNA and RNA, the first one is that their sugar molecule is different because DNA has deoxyribose, RNA has a ribose. Their nitrogenous bases are different, just one. The first one is adenine, that one is the same, AA. The second one, DNA has thymine and RNA has uracil. The third one is the same, cytosine, CC, and then guanine, GG. Then you don't forget that with DNA, adenine always pairs with thymine and vice versa. Cytosine always pairs with guanine, GC or CG. Okay. With RNA, adenine pairs only with uracil, and then cytosine also with guanine. Then their strand, DNA has a double strand, DD, and then RNA has a single strand. I'm going to put the picture up again for you. Let's tackle just a little question. I've got 4 more, but I'm just scrambling through the ones that are dealing with this topic only. Okay, I found this one, which is in section A, question 1. So it reads, which one? So I'm going to be highlighting also the important words to look out for. So I'm looking for one. It's only one option that they want. Which one of the following correctly describes a difference? So I want a difference between DNA and RNA. Now you don't forget that we just did our notes, which states the difference. Let me write that one. Differences. Okay. Now we want something that is different between DNA and RNA. Okay. So instead of just scrambling and just looking for the answer, we need to analyze every option we are given and just take out the ones that we know are not true. First one, A, says DNA has a deoxyribose sugar, RNA has a ribose sugar. This one is an option. Second one says DNA has uracil. No. RNA has thymine. No. This one is just out. Okay. Third one says DNA has a single strand. Because we remember DD, DNA double. So that one's out. RNA has a double. No, that one's out. We go to the last one. DNA has a different number of cytosine and guanine nitrogenous bases. That is not true because if they are pairing, they should have the same number. Let's see, RNA has the same number of cytosine and guanine. Yeah, that one is true, but that one is not. And also, it's not now the problem with this one. Yes, this statement shows the differences, but the nitrogenous bases are not different. So that option is totally out, because these nitrogenous bases are the same and you are looking for a difference. So there is your first answer there. There's the answer. So please go through all of them and try to, take the other ones out. Therefore, my answer there would be A. This is another section A question 1 question because of this numbering 1.1.8. Right. The table below shows percentages. So we are dealing with percentages. I like to use the red pen because it's different from the question ink, which is black. And then I would usually answer with blue. Right. Okay, so we're dealing with percentages of bases, which means nitrogenous bases. In DNA, we are focusing on the DNA one. And you remember from your notes that with DNA, here are your nitrogenous bases: adenine, thymine, cytosine, and guanine. And remember, I just said now, because they pair together, they should have the same number or the same percentage. Everything should just be the same so they can pair nicely. All right. So again, percentages of bases in DNA. Right. Now we look, source of DNA, human liver, adenine 30. Therefore, I look for the one that it pairs with. Thymine should also be 30. I start there in the question before even going to the option. You try to fill in and try to see what you remember. Now, adenine will always pair with thymine, so they should be 30-30. And then cytosine with guanine. Okay? With chicken liver, adenine is unknown, but I know that thymine is 10. Therefore, it should also be 10 because they pair together. And these ones are fine also. Dog liver, adenine is 24. I check the thymine. Ooh, okay, that one is just not working. Even these ones are not working. Okay, let me leave that one out. Mouse bone marrow. Okay, adenine is 23. Thymine is that. Okay, leave it out. Anyways, they want me to fill in these ones. Let's see. If guanine is 26, cytosine will be, yep, you guessed it right, 26. They should be the same. Adenine in the sun leaf is 10. Thymine is 12. Definitely out already. Okay. And then cytosine, if it's 39, it pairs with guanine, which will also be 39. All right. Let me go now. Which row of the table below correctly reflects the missing data? Okay. I know now. Right. W should be a 10. So I'm going to look for a W, which is a 10. Therefore, these are my options. That one's out. Okay, X should be a 26. Okay, so these are my options down to 2. Y should be a 39. Okay, I've still got 2 options because that one's out already. Okay, because of those. Z should be a 30. Aha, there it is. Therefore, my option there is a C. All right, for the third question, it's 1.5, so it's also section A. Okay, the diagram below represents some nucleotides. So remember, your nucleotide there is your monomer in a single strand of DNA, right? So this is DNA. Now, you know DNA has a double strand, but here they're just showing us the single strand. So please don't even affirm to this trick. DNA, it's double. They're just showing you the single one. Okay, here it is. You just go through it. This is your circle, which means your P. I can already put here. I know they want that one. So this one is my P, my phosphate. This one is your sugar. Okay, there. So it's my sugar. But then, because it's a DNA, what sugar is this? Right, you guessed it. Deoxyribose. Right. And then these ones are my nitrogenous bases. Okay, let's try to remember. T for thymine, G for guanine, A for adenine, and C for cytosine. Okay, let's go to the question. 1.5.1. Write the letter. They even put it in capital letters for us so that we A, you must remember. Just the letter of the part that represents a sugar molecule. Which part represents a sugar molecule? You know what? I'm going to get a paper for this and write it nicely. Okay, because there's a lot of questions now. Let's see. So we've got 1.5, then we've got 1.5.1, then we've got A. Okay, let's go back. Write the letter of the part that represents a sugar molecule. So they want the letter. Okay, there's my sugar molecule there. Therefore, it is just a Y. Okay, next one, they want the letter of the part that represents a phosphate. There it is. It's my X there. So please don't now be putting that P that you just wrote. That one is just for you to remember. Okay, we'll go to the next question. How many? They want the number. How many? So how many nucleotides? Then you remember, okay, if it's a nucleotide, let me just use, you know what, you are allowed to use your question paper. This is your baby. You need to remind yourself here. Let me quickly do it here. Okay, so a nucleotide, she said it's please. Okay, even if you draw it wrong, it's fine. Stop. Okay, let me remember. Please stop. All right. Now, babe. Okay, so this is my nucleotide. Okay, it should look like this. Because obviously, I mean, you're in the exam, so you cannot be making noise and you're just trying to remind yourself, which you are allowed to do. So here's your nucleotide. It's one. This is one nucleotide. So here, if they're asking how many nucleotides are represented in the diagram, that one. Now we are going to count, okay? This is 1. This is 2. This is 3. And 4. There you go. There's your answer. Be confident with it also. Okay, that's 1.5.2. It is 4. You can write it in words also. Next question. Write down the nitrogenous bases. Okay, nitrogenous bases, these ones. Okay, from top to bottom. Okay. Okay, from top to bottom. Top to bottom. There's even an arrow as indicated by the arrow of the complementary DNA strand. So they want the ones that will complement those ones. You remember? Because these ones are given. What will complement? So from the top, then you say, all right. 1.5.3, you can write it whichever way. You can tell them T will be complemented by that, or you can just write the answers. I'll just do that. The first one will be A because T always is joined by an A. And then G will be joined by a C, which is cytosine. A again is joined by a T. You remember now? AT, TA, GC. And then last one, C will be complemented by G. So that is how they will be complemented. And also, if you want to just make sure, you can put it there. AT, okay, GC, AT, CG. There you go. There it is. Right. Last one. Name 2. Okay, two of them. Processes. Ooh. Okay, that require the 2 strands of DNA, the natural ones, DNA molecule to separate into a single. All right. Now, these notes we have not got into. It's for the next video, but we can answer it because remember, this is revision. So you should at least remember. Right. What are the 2 processes that will cause my DNA strand to separate? Those ones, first of all, it's the DNA replication. You remember I said the fact that your DNA strand has weak hydrogen bonds is so that it can unwind. So it's DNA replication. And then the second one is protein synthesis under that first sub. Then for our last question of this video, we're doing 2.1, which is in section B, question 2. Okay. The diagram below represents one strand. So it's just one, again, which is given one strand of a DNA. It's for a DNA. So make sure you highlight that for yourself so that you don't think it's an RNA. Here it is. Here's the one strand, okay, with the one nucleotide, 2, 3, 4, 5, 6, 7, 8, 9. Okay, that one has been circled for me. That tells me something. and I've got a Y there pointing right here. Then I remember the circle is also always P. That one is the sugar. Okay, let me write it there. There's the P, and then there's the nitrogenous base adenine. Then I come here again. Here's my phosphate. There's the sugar. There is the nitrogenous base guanine. And remember, this sugar is a deoxyribose because you've got a DNA, right? Now answer it in a paper also. Okay, let's see. 2.1.1. Sorry, let me start with 2.1 right there. And then A. Okay, they want me to identify sugar Y. That one. Then I remember that because it's a DNA molecule, sugar Y is a deoxyribose sugar. Okay, I go to B. B wants me to identify molecule, a full structure, Z. That full, what's that? That one is a nucleotide. All right. That's it for today. So these were the notes we're using today. I will try and put them up so that you can have a nice screenshot of them. And also I said, if you want them, I can just hook you up, but I'm going to probably scan them or what, and then all these colors will be gone. Oh my word, I love my colors. It was so nice being with you guys. Please don't forget to share and subscribe. It's so that we can get our notification bells. Guys, I appreciate all comments. Also, I'm novice, so let's grow together.